honored to be here. I want to thank Dr. Seckin not only for bestowing this tremendous honor on me, but also for founding this fabulous society that he and Padma put together that I think will be a big boon to the field and to women. Today I'm going to talk to you about a few different topics, some of the research that we've done over the last several years on endometriosis, and I hope you'll find it interesting. Some exciting new findings both in genetics, epigenetics, and the role of stem cells in endometriosis. First, what is the pathogenesis of endometriosis? And an age-old question that's in every introductory textbook on obstetrics and gynecology. We'll learn about the theories for the origin of endometriosis. And the most commonly accepted theory for the origin of endometriosis is retrograde menstruation, or Sampson's theory. But we've heard of the theories of embryonic rests and solemic metaplasia. Clearly, as others have pointed out this morning, there is certainly a familial tendency to develop endometriosis, so there is a genetic component, and we'll talk about that today. And we'll also talk about the role of stem cells in endometriosis, something that we've been working on for the last several years. So why do we think, though, that most endometriosis comes from retrograde menstruation, consistent with Sampson's theory? Well, the endometriosis tends to most commonly be found in the dependent portions of the pelvis where you'd expect it to settle as a result of gravity. We see retrograde menstruation in almost all women, so we know those cells get there. We see a very high incidence of endometriosis in women with the obstruction of their outflow tract. If the cervix is somehow constricted, more retrograde flow, and if you have no cervical patency, you'll almost inevitably get endometriosis. So we know that enough bulk of flow in a retrograde fashion certainly does lead to endometriosis. Also, the tubes are often patent, even some of the worst cases of endometriosis where we see a lot of adhesions. Remarkably, the tubes are often still patent, allowing that retrograde flow. And the risk factors that we heard about earlier this morning, frequent menstruation, early menarche, and probably most importantly, the animal models that Dr. Hugh and others have championed, that even placing normal endometrium in the peritoneal cavity can lead to endometriosis. You don't need to have defective endometrium to begin with to get endometriosis. Yet I don't think Sampson's theory really explains all of the endometriosis we see. It certainly doesn't explain endometriosis outside of the peritoneal cavity. How does endometriosis get to those areas like the brain or the lungs, where those of us who treat a lot of patients with endometriosis have certainly seen it? Nor does it explain endometriosis after hysterectomy, nor does it explain those rare reports of endometriosis in men, typically men being treated for prostate cancer and receiving estrogens. Rare, but certainly that does not come from retrograde menstruation. So there must obviously be other etiologies for endometriosis. And first, I'd like to talk to you about our stem cell theory for the origin of endometriosis. First, what is a stem cell? I think many of you in the room are probably already familiar with the concept of stem cells. It's a relatively undifferentiated cell that can not only replicate itself, but that can give rise to more mature cell types. You know, when most cells divide, the two daughter cells are identical to the parent cell. But a stem cell replicates itself in an undifferentiated fashion and then gives rise to these other progeny that can differentiate into more mature cell types. And we have these very undifferentiated stem cells that self-renew, that are relatively quiescent, don't multiply much, and can reconstitute tissues. They give rise to local progenitor stem cells that can give rise then to all of the specialized tissue in a particular organ system, and they go through this phase of these transient amplifying cells that are, again, much more restricted and not in this undifferentiated state. So there's a hierarchy of stem cells. And the endometrium, we've known, even before there was a concept of stem cells, that this highly regenerative tissue that undergoes complete renewal in each menstrual cycle probably has some sort of cell in the basalis layer of the endometrium that can give rise to a new endometrium every menstrual cycle. And again, even before we had a concept of stem cells, we all had some theory about these regenerative cells in the basalis layer of the endometrium. And indeed, some of Carolyn Gargett's work, who was the honoree here last year, identified some of these progenitor stem cells that are in the endometrium and can regenerate the endometrium in each menstrual cycle or estrous cycle in a mouse. So it's probable that most endometriosis that arises through retrograde menstruation comes from these progenitor cells, these cells in the endometrium, when they're shed in a retrograde fashion, and then can regenerate an entire endometrium or an entire endometriosis lesion within the peritoneal cavity. But over the last 20 years, we know that there are multipotent stem cells that are found in multiple tissues, and they have a vast ability to differentiate into a huge number of cell types. They are multipotent. They're rare, but some of them are found in the circulation, so they can move around the body and relocate to new places. So the first thing we asked is, could these stem cells from other locations potentially engraft the uterus? Could these be the cells that can repair and restore the uterus in the event of injury or repopulate the endometrium in the estrous cycle or menstrual cycle? The first place we looked was the bone marrow, and the bone marrow is known to have a large number of these stem cells. Not only stem cells that give rise to blood cells, the hematopoietic stem cells that we commonly think of, but also a mesenchymal stem cell that has the ability to give rise to a vast number of mature cell types, including fat, muscle, and epithelial cells, for example. We reasoned that this might be a source that endometrial stem cells could be derived from. If bone marrow could give rise to all these different sorts of cells, certainly they should be able to give rise to endometrium, which has this tremendous need for regeneration. So we asked whether bone marrow-derived cells could repopulate the endometrium, first in a mouse model. And here you can see we looked at Y chromosome fish. So there's fluorescent in situ hybridization. The red dots are Y chromosome. We took male bone marrow, transplanted it into female mice, and then looked for Y chromosome cells in the endometrium. A here is a control male tissue. You can see most of these cells express Y chromosome or show a signal for Y chromosome. B is the uterine tissue, an endometrium from a mouse that got a female bone marrow transplant as a control. No Y chromosome signal. But here in C and D are the mice that got transplanted with male bone marrow. These are endometrial sections, and here's the endometrial lumen, here's the endometrial epithelium, and here's a Y chromosome bearing cell in the endometrial epithelium of this uterus after bone marrow transplant. And here similarly is a stromal cell. Now, after bone marrow transplant, you'd expect all the white blood cells, of course, to be derived from the bone marrow donor, just to make sure we weren't seeing some of the white blood cells that would naturally migrate into the uterus in each cycle. We also looked at CD45, which is a marker for all white blood cells, and we looked at some other specific epithelial or stromal cell markers. Again, here's our male as a control. Here's a white blood cell. This is not what we were looking for, or this is what we'd expect in these. This green is showing CD45, a marker of leukocytes, and it's got a Y chromosome. But here in this panel, you can see the cell that has a Y chromosome. It's not green, so it's not a white blood cell, and it expresses cytokeratin, which is a marker of epithelial cells. So that's an endometrial epithelial cell that came from that bone marrow. It's a male cell that is now in the endometrium. Well, that's a mouse model. Could this happen in humans? So we asked if women who'd had bone marrow transplants also could have some of these stem cells that eventually are incorporated in their endometrium. And we looked at just a small number of women who'd had a bone marrow transplant with a single HLA antigen mismatch that allowed us to identify the source or origin of any cell. They were reproductive age women. They'd all had chemotherapy and total body irradiation, followed by bone marrow transplant. And this is what we saw. The brown here is standing for the mismatch HLA antigen, that of the bone marrow donor. And you can see that some of these cells are intercalated here in this epithelial lining. The blue are the endogenous cells, and some look like they have the mismatch HLA antigen from the bone marrow donor. We looked in the stroma, and here we serial stained for the CD45 to look at leukocytes and also the mismatch HLA antigen. These red cells here are the ones that are staining for CD45. Those are the leukocytes that you expect to be all of bone marrow donor origin, of course. Somebody had a bone marrow transplant. The blue cells are the cytoplasm of those cells are not staining. There's just the nucleus you see there. Those cells are the endogenous stromal cells. But the brown cells that the arrows point to, these are the cells staining for the mismatch HLA antigen of the bone marrow donor. They are not white cells. Those are the stromal cells that are of bone marrow donor origin. So again, in humans, we see stem cells going to endometrium. So stem cells are recruited to the uterus for repair and regeneration of the endometrium. But of course, we really want to know, could stem cells contribute to disease, such as endometriosis? So again, we created a mouse model of endometriosis. We sutured pieces of the uterus into the peritoneal cavity to make experimental endometriosis in mice. And we used two different strains of mice, wild-type mice and LAC-Z transgenic mice. And the LAC-Z gene we could just use here as a marker, again, to tell us the origin of any given cell. The mice had a hysterectomy, so none of the cells that we saw migrating here were from the uterus. They could not be from retrograde menstruation. They could not be from hematogenous or lymphatic spread of endometrial cells. And we looked at then the expression of this LAC-Z gene in our endometriosis implants. So to set this up, this is our wild-type control mouse uterus, or endometriosis rather. No staining for this LAC-Z gene. Here's the LAC-Z transgenic. Every cell stains brown. So those are the LAC-Z transgenics. And when we transplant the wild-type endometrium into the peritoneal cavity of the LAC-Z transgenics, we get something that looks like this, wild-type, and here's the demarcation where we have the border between the endometriosis that we've created and the peritoneal cavity. So what did we find? Well, after some time, we started to see cells that were incorporated into the endometrium, endometrial cells, that were LAC-Z expressing cells. So these cells came from a source other than the uterus. These are stem cells that differentiated here into stromal cells. And this, again, is looking for immunologic staining of the LAC-Z beta-galactosidase gene, or protein that's encoded by the LAC-Z gene. Just to make sure this wasn't an artifact of our immunohistochemistry, we also looked for enzymatic activity of that gene. The beta-galactosidase activity turns a certain substrate blue. And here you can see this is the uterine lumen. This is an epithelial cell, and this is a stromal cell. So these cells come into this endometriosis, are not from a uterus. Remember, these mice had a hysterectomy. These cells are migrating into the endometriosis and driving endometriosis. So this is a novel origin of endometriosis. Stem cells contribute to murine endometriosis. Ectopic stem cell differentiation can cause endometriosis and likely accounts for the endometriosis outside the peritoneal cavity and perhaps endometriosis after hysterectomy. So even when we do a very complete job of excising endometriosis or even a hysterectomy, how do we still get endometriosis? Well, this may be one mechanism. And more than that, I think this is a novel mechanism of disease. Ectopic transdifferentiation of stem cells may indeed lead to other diseases. Endometriosis may only be the first example. Just so you don't think this stem cell migration is all a bad thing, the fact that the endometrium and the uterus has these multipotent stem cells we can use for various therapeutic purposes. And I'll just go over this briefly as an aside to show some of the benefits of these stem cells. We can sort these cells, and I won't go over all the markers that we use here, but we can use FACS sorting to pull out the stem cells from the endometrium. And then we can use various protocols that we essentially borrow from the embryonic stem cell world, and we can differentiate these cells into all sorts of different mature cell types. Here we make chondrocytes, cartilage cells. Here we actually got them to differentiate into neurons. And whether their potential to differentiate into neurons may play a role in the nerve fiber seen in endometriosis patients' endometrium is still a question. But they look like neurons. They connect and synapse with the surrounding cells. And when we create a Parkinson's disease model in mice, we got them to produce tyrosine hydroxylase, which is the rate-limiting enzyme involved in dopamine production, and we create Parkinson's disease in mice, and we transplant these cells in versus a control, we can help rescue dopamine production in these mice. So here are the unlesioned animals. Here's what happens when we give them this medication that causes Parkinson's disease. We transplant our endometrial-derived stem cells into these mice, and their dopamine production goes back up, helping to partially rescue Parkinson's disease in this animal model. So these stem cells from the uterus have tremendous therapeutic potential. Finally, more recently, we were able to differentiate these cells into insulin-producing cells that resembled pancreatic beta cells. Here's our culture. The green here is standing for insulin. There's our controls up here. Here are our insulin production after beta cell differentiation. And not only do these cells make insulin, but in culture, when we treat them with glucose, they make more insulin. So they're glucose-responsive insulin production, resembling exactly what we'd want to see from a pancreatic beta cell. Here's the undifferentiated cells, don't make insulin. Low glucose, they make a little. High glucose, they make more. When we transplanted them into mice under the kidney capsule, you can see that several of these cells continue to produce insulin. And when we create a diabetes model in mice, here are our glucose levels. These are our untreated. Here are our controls that did not receive stem cells, received undifferentiated endometrial cells. And here are the mice that received stem cells from the endometrium that we differentiated into insulin-producing cells. Their glucose control was better. More dramatically, those were fasting levels. We think we probably evened out more of the peaks of glucose that we see after meals. More dramatically, these mice look dramatically different. The mice that were treated with our stem cells weren't dehydrated, they were active. The mice that were not treated with stem cells, that were treated with control endometrial cells that weren't differentiated into insulin-producing cells, lost weight, became dehydrated. Most of them developed cataracts. They were lethargic. You could easily tell from across the room. So the good, the bad, and the ugly of stem cells. The stem cells are there to help repair the uterus and regenerate the endometrium in each menstrual cycle. They can repopulate the endometrium, repair the uterus after injury, but they also cause disease. They can contribute to endometriosis, again, accounting for some of those more unusual forms of endometriosis. But they also have tremendous potential as a new and easily accessible source of immunologically matched stem cells for women that can be used for tissue engineering and regenerative medicine. I'm going to shift gears a little bit now and talk to you about some of our findings that we just published a couple of weeks ago on a genetic basis for endometriosis. So if all women have retrograde menstruation and all women have these stem cells, why do only some women get endometriosis? And why do we see an increased risk of endometriosis if someone has a mother or sister with endometriosis? Clearly there is a genetic component, and many, including some people in this room, have been working on this. But one of the best candidate genes as an endometriosis, perhaps causing endometriosis, is the KRAS gene. And another group several years ago published a paper showing that an activating mutation of an oncogenic KRAS gene leads to endometriosis in mice. And this is really the only de novo endometriosis model in mice. We can transplant endometrium into the peritoneal cavity and create experimental endometriosis. But mice with this genetic defect develop spontaneous endometriosis. Well, is this a novel genetic etiology of endometriosis? Well, a lot of people started looking then for mutations in the KRAS gene in women with endometriosis. And despite an extensive search and several publications, no one's found one. But we hypothesized that there may be a defect not in the KRAS gene itself, but maybe in some of the regulators of KRAS expression. And interestingly, KRAS is regulated by a microRNA termed LET7A in particular. For those of you who are not familiar with microRNAs, these are small areas of the genome that don't encode protein. They're transcribed, they're cleaved, they're exported from the nucleus. They are then small, about 20, 22 nucleotide, single-stranded RNA pieces that are incorporated in this RISC complex. They bind to other genes where they have complementary base pairs, and usually in the non-coding region, often in the 3' untranslated region of a gene. And when these small RNAs bind, they usually either lead to decreased stability of the RNA or block translation. So you get less of the gene produced. So these microRNAs, very common, and they regulate genes. Well, the KRAS gene has a binding site for this LET7 microRNA in its 3' untranslated region, not in the coding region of the gene, but in this area that controls RNA stability and translation. And when LET7 binds, it blocks translation of the KRAS. You get less KRAS protein. Well, we wanted to look to see if there were potential mutations in this LET7 binding region. Some polymorphisms in this region had been described in humans. So we screened about 150 women with endometriosis for polymorphisms in this LET7 binding site in the 3' untranslated region of the KRAS gene. And we found that about 30% of the women with endometriosis, now this is my patients with endometriosis, so it's a referral practice, more commonly women who've failed treatment with oral contraceptives or been referred from other gynecologists. So maybe women with more severe endometriosis, but about a third of them almost had this KRAS variant, as opposed to only about 5% in a large, diverse population. And Idalis Flores is in the audience here, was a collaborator with me on this study. Many of the patients were hers. Not just my referral population, mine and Idalis's population. So what happens when they lose this binding site? Again, here's the LET7 binding site. When you mutate that binding site, the LET7 doesn't bind. You get an A to G mutation. LET7A can no longer bind. We look to see if that did indeed result in increased KRAS expression. When you have the loss of that LET7 binding site, we compared either normal women without the KRAS mutation to endometriosis patients who had a little bit higher KRAS expression, but we looked specifically at the endometriosis patients with the KRAS mutation. The expression of KRAS RNA was dramatically increased. And indeed, same thing, the KRAS protein was significantly increased. To make sure that this really was this mutation that was regulating that, not something else about the cells from these women with endometriosis, we took out just that 3' untranslated region of the KRAS gene and made a genetic construct where we could put it in another cell type and just see what happens to KRAS expression or a reporter gene that's linked to that 3' region in association with either the LET7 or endogenously without the LET7. So again, we have a reporter gene, a gene that just sends out a signal that lets us know the influence of this mutation or not mutated site in a cell type unrelated to endometriosis. So we know this isn't just some other artifact of the endometriosis cells themselves, that it's specifically this particular site that is causing the change in expression of KRAS. And when we look at the wild type KRAS 3' UTR in this assay, fairly low expression. When we put the variant in with the mutation, the signal goes way up, showing that loss of that signal really drives gene expression up higher. When we now add a new microRNA that's designed specifically to bind the mutated site, to mimic what the LET7 was doing on the original site, now on the mutated site, we drive the expression of this KRAS back down. And a control siRNA that doesn't specifically bind that site doesn't do it. So it looks like that mutation really is driving KRAS expression. We also looked at what happens then to all the LET7 microRNAs. I told you LET7A normally binds there, but there's a whole family of LET7 RNAs shown here. And in women with this mutation, they all had much lower expression of all of the LET7 microRNAs. Now, LET7 doesn't just bind KRAS. It binds a whole lot of things and regulates a lot of different genes. And again, when these microRNAs bind, they generally downregulate genes. So when LET7 is lower in women with this mutation, it's no longer stopping the expression of many genes that are involved in mitotic signaling, not only RAS, but genes like MYC. It's no longer stopping things involved in cell cycle, like cyclin-dependent kinases and cyclins, and other genes that are involved in cell adhesion and migration. there may be more effects other than just those based on KRAS. And we looked indeed at how these cells behaved. We looked at BRDU incorporation, which is a measure of how quickly they proliferate. The ones with the mutation proliferate faster. We looked at their ability to invade extracellular matrix. How invasive are these cells? And again, those from endometriosis are a little more invasive, but those with the mutation are the most invasive. So it does seem to change the behavior of these cells. Then finally, we took cells from women with or without the mutation, and we transplanted them under the kidney capsule of immunodeficient mice. And they both formed lesions that look like endometriosis. But those with the mutation expressed a gene called PCNA a little higher, which is a gene involved in proliferation. It tells us these cells are proliferating faster. And they had lower expression of progesterone receptor. And progesterone resistance, as you know, is a characteristic of some women with endometriosis. So this is the only clearly identified so far genetic cause of endometriosis. And knowing a gene responsible for endometriosis may allow us to do some sort of risk assessment, especially in families where we know that this gene is the etiology of endometriosis. Again, it's not found in all women with endometriosis, so it won't be a general screening for endometriosis, but in those who we know have KRAS-related endometriosis, it may allow us to screen family members and assess risk. It may be that different pathways are affected in this particular mutation. So it may allow us to individualize or personalize treatment. Then we may find that these particular variants of endometriosis respond differently than others. And it also provides novel targets for treatment. And right now we're doing experiments looking at altering some of the pathways that I showed you here to see if we can develop novel treatments for endometriosis. Other groups have done studies looking for genetic etiology of endometriosis, and again, some of the people in this room have been involved in these studies. Some of the larger screening studies have been the genome-wide association, or GWAS studies, that have tried to equate or correlate single nucleotide polymorphisms, or those normal genetic variants that we all have, with disease. And one of the best studies, a large international consortium, identified one candidate gene that had a odds ratio of about 1.2, almost 1.4 for severe disease, was a SNP that was located upstream of the HOXA10 gene. Now, those of you in the audience who know me know I've been working for the last 20 years on the HOXA10 gene. So this was music to my ears when I heard that this gene that we had associated with endometriosis may really be genetically associated with endometriosis as well. For those of you who are not familiar with this, this is some of our work. We showed during the menstrual cycle in the uterus, this gene, expression of this gene goes up. This is early mid-proliferative phase, early mid-secretory and late secretory phase. It goes up around the time of implantation. When we finally got a good antibody to it, you can see it's the glands that start expressing this right in the middle of the window of implantation. So that's what really, it's the glandular expression that goes on that accounts for that uptick at the time of implantation. And if you knock this gene out in a mouse, they are infertile. The uterus is defective. Implantation won't occur. The embryos are fine. If you take them out and put them into a surrogate, they do just fine. But if you put even a normal embryo in that uterus, it won't implant. So it's necessary for fertility. And we showed some time ago that in the endometrium of women with endometriosis, you don't get that same cyclic increase in the expression of this gene. The red here, the blue shows the women, this is across the x-axis is the menstrual cycle. The blue shows an increase across the menstrual cycle that I showed you earlier. The red shows the women with endometriosis. You fail to get that upregulation at the window of implantation, which I think accounts for some of the infertility we see. So why is this gene expressed at a lower level? Well, we turned to some of the animal models of endometriosis, both mouse and non-human primate. And when we create experimental endometriosis, we put endometrium in the peritoneal cavity, and we look back at the expression of several genes that are important for implantation in the uterus. They're affected. They didn't have an abnormal endometrium to begin with. These are mice that were completely normal, but just placing normal endometrium in their peritoneal cavity changes the gene expression in the uterus, including that of HOXA10, the gene that I showed you earlier. In baboons, some work we did in collaboration with Ozgi Fazlibaz, Again, we can transplant endometrium into the peritoneal cavity, get lesions that look just like human endometriosis. And you follow out the expression of this HOXA10 gene, and it goes down. It takes a while. This is months. It took about a year before we saw a significant drop. But of course, most of our patients have probably had endometriosis for years before we diagnose it. But it looks like endometriosis is having an effect on the endometrium. You don't have to have a genetic abnormality or abnormal endometrium to have an effect. So transplantation of even normal endometrium gives rise to endometriosis, and that in turn affects gene expression in the uterus. How does that happen? Well, now I want to talk about the third topic that I promised I'd get to in my title, epigenetic alterations in endometriosis. It's not just genetic. Changes in DNA, other than simple mutations, can influence gene expression. We now know that DNA is very highly modified. CG base pairs, or cytosines in the CG base pairs, can be methylated. Histones, which package the DNA, can be methylated, acetylated, phosphorylated. Many covalent modifications change the packaging of DNA and change gene expression. Well, the HOXA10 gene that I showed you has a lot of these areas that have multiple CG base pairs clustered together, or so-called CG islands. So we wondered if, in the presence of endometriosis, these might be methylated, and that might explain the lower expression of this gene. Well, indeed, we looked in our mouse model, and in particular, some CG base pairs in the promoter region were very highly methylated compared to the controls. So it looked like that methylation was indeed suppressing the expression of this gene in endometriosis. In our baboon model, the top boxes are the promoter region of this gene, so-called F1. This top block here is those disease-free animals that were our controls that did not get endometriosis. Here are the animals with endometriosis. The promoter region of this gene was the only area that was methylated differentially, and it was much more highly methylated in the baboons after creation of endometriosis. So it shows that transplantation of even normal endometrium into the peritoneal cavity leads to methylation, decreased gene expression, and Sunway Gao's group in Shanghai has also shown that the same methylation pattern is seen in humans with endometriosis. So increased methylation represses HOX expression. This expression, you can get there probably a couple different ways. Anybody with endometriosis is going to have some methylation and epigenetic repression, but based on the genetic studies I showed you earlier, maybe there are also some people that are born with a genetic propensity to have lower HOX expression, or an altered regulatory region of this gene may be one other etiology of low expression of this gene. This gene is necessary for normal differentiation of the endometrium, lower in women with endometriosis. So multiple different ways to get there, but probably very similar end results. So let me end by turning to our clinical treatments. Can we treat infertility associated with endometriosis, and how do we treat it? Well, medically, I think we all agree that there's really no role for medical therapy for endometriosis. Multiple studies that have looked at various drugs for treating endometriosis-related infertility have shown no significant improvement, so we don't use various medications for infertility treatment. They may work well for pain. How about surgical therapy? Well, there have been a couple of large prospective randomized trials, the larger being the ENDOCAN study. And indeed, surgery does improve fertility. This looks at weeks after randomization to either surgery for ablating or resecting the endometriosis versus a diagnostic laparoscopy. And the pregnancy rate goes up considerably when we actually remove the endometriosis rather than just look at it. But you'll notice that the control group had about a 2.5 pregnancy rate per cycle. And yes, we can double the pregnancy rate to about 5% per cycle, but it doesn't go back to normal. So why not? Why don't our treatments work better? How does endometriosis affect fertility? Well, it may be that we can be a great surgeon and remove all of the endometriosis, but based on the methylation that I showed you, based on some of the epigenetic changes that I showed you, you remove all the endometriosis, you may have helped or prevent pain, but the changes in the uterus are left. These epigenetic changes are very slow to turn over. Maybe that they do in time, but they're not turning over rapidly. So even when we do a perfect job with our surgery, we may be left with some residual endometrial epigenetic changes and maybe not a full cure. So even when we take all the endometriosis out, it could come from stem cells. Even when we do a hysterectomy, it could come from stem cells and find its way back into the peritoneal cavity or elsewhere. Or even when we take all the endometriosis out of the peritoneal cavity, we may be left with some residual epigenetic defects that can still cause infertility. So I'll summarize by saying endometriosis probably isn't just one disease. It's a syndrome characterized by not only genetic susceptibilities and genetic forms of the disease, but also cellular that may be related to stem cells and epigenetic alterations that result in this ectopic localization of endometrium and defects also in the eutopic endometrium. So endometriosis certainly has a genetic predisposition, and we think we found the first genetic cause of endometriosis in this KRAS variant. Stem cells contribute to both endometrium and endometriosis, and the endometriosis induces certain irreversible epigenetic disease changes in the endometrium that may make this disease not fully curable. We can get rid of the lesions, but there may be some residual effects left behind. So I'll summarize by saying endometriosis, most of it probably comes from retrograde menstruation, but it also can come from stem cells. Everyone has both of these, so some people probably have a genetic predisposition, either this KRAS LET7 binding site or maybe a HOXA10 mutation that was found in the GWAS study. Those probably have lesions that are more prone to proliferation and invasion. They may be more likely when they have retrograde menstruation to develop endometriosis. And anyone with endometriosis then develops a defective endometrium, and the epigenetic alterations may very well mimic some of the same genetic defects that some people are born with. Of course, the environment can influence these, the medications we use, some of the exposures that were mentioned earlier may all impact on this process, and that tells us also that we have the ability to modify the process and perhaps improve things for our patients. So I'd like to end there. I want to thank some of my collaborators at our Yale Center for Endometriosis, where we do much of this research, some of the people in my lab who have been involved in the endometriosis research. There's some other people working in the lab that are working on other projects, so don't feel slighted that you're not mentioned there, Leo. Some of my collaborators that have done some of the animal studies with me, and the NIH, of course, for financial support. Thank you.
Thank you, Professor Taylor, for this